Hello everyone. Now let's proceed to see test of heterogeneity during meta-analysis. So in meta-analysis, the most important concern that we need to deal with is whether there is a heterogeneity between the research articles that have been included for our meta-analysis result. So first and the most important way of testing heterogeneity is by the use of a Galbraith plot. So let's see how to do this Galbraith plot in order to test whether there is heterogeneity between the included studies for our meta-analysis research, using a software which is Stata. So in the Galbraith plot, it is really used to assess whether there is heterogeneity between the studies and to detect potential outliers, because if there is a divergent result or finding between research articles that have been selected for meta-analysis, and if there are either extreme large effect size or extreme small effect size among the outputs or the results of the studies included in the meta-analysis, that could be made possible to detect by the use of a Galbraith plot. So that heterogeneity is because of the result of potential outliers among the included studies. So the way that we need to test this heterogeneity because of potential outliers is detected by the use of a Galbraith plot. That is a really very fundamental. So it may be also used to, or an alternative for forest plot, for summarizing meta-analysis results, especially when there are many studies. When there are many studies, it could be just used as an alternative method for forest plot in order to summarize the meta-analysis result. In addition to just detecting potential outliers and heterogeneity between the studies, it could be used for summarizing the meta-analysis result. So now in this video, we are going to just use an example dataset which is based upon by using a BCG or the BCG vaccine dataset. So the BCG vaccine was just given for one group, that is a treatment group, and there's another group which is a control group. And then the number of TB positives and TB negative individuals were reported in both the control and the treatment group. So among the included studies, whether there is heterogeneity in terms of the effect size of BCG in preventing tuberculosis was just aimed to evaluate whether there is any potential outlier and heterogeneity in terms of the effect of BCG on prevention of tuberculosis. So this is an example dataset that we are going to use, or we are going to use for this demonstration. So after just running the Galbraith plot, the output will look like this one. So we need to understand what does this Galbraith plot tell us. We need to interpret it correctly. What is the interpretation or the understanding or the message of this Galbraith plot, which could be produced by the use of different research outputs which have been included for meta-analysis. So what does it mean? After we run a Galbraith plot, we will just obtain such a graph, or simply a Galbraith plot. So in this plot, we see that the blue dots here are the individual effect sizes of a drug for each study. Within each study, what does the effect size of an intervention on the disease? So study-specific effect size is reported by such circular blue dots are indicating specific study effect size of an intervention for the disease. So that is the result of each specific study is indicated by the use of these blue dots, the blue dots. And then this is a 95% confidence interval. The shaded area is the 95% confidence interval for those included studies in the meta-analysis. And then this horizontal line, which is drawn at zero level. In front of zero, There is a 0 score, and above 0 there are positive numbers, and below 0 there are negative numbers, you know. So the horizontal line which is drawn in front of this whole 0 score is indicating that no treatment effect. No treatment effect. And this line is a regression line. This is the slope line. This one. The line which is in within the shaded area, the confidence interval. This is the regression line, you know. The overall summed effect or slope of the effect of the treatment on the disease, which was found or recorded from different research articles which have been selected for meta-analysis. So this is a regression line indicating that the effect of an intervention on the outcome. That is the regression line. And the shaded area is the confidence interval. But this horizontal line, which is drawn in front of 0, is said to be no treatment effect. No treatment effect. Okay. So now here is again a very important idea, you know. Those studies, for example, this dot and maybe this dot also, those studies which are above the reference line or the no treatment effect line indicates that there is an increasing risk on the treatment group. Individuals which are taking the drug are higher risk of developing the disease, you know. And the dots which are located below this no treatment effect or no effect, so these dots which are below this line are said to be indicating that there is a reduction of disease development among the treatment group. So the treatment groups are being protected from developing a disease because of taking the drug. So that the dots or the blue lines which are below the no treatment effect line, below this line, are indicating the drug is protective of developing the disease. So that is really having an intervention effect, you know, or a prevention effect from developing the disease. So that dots which are above the no treatment effect indicate that there is a higher risk of developing the disease because of taking the drug. And the dots which are below this no treatment effect line are indicating the drug is effective in preventing the disease. There is less risk of developing the disease in the treatment group. Okay, and another issue that we need to understand here is the blue lines, these dotted, the dots or specific studies results which are near to or close to the y-axis are said to be less precise. Less precise. That means the confidence interval is wider. For those dots, these dots which are close to the y-axis are having less precision. That means they do have wider confidence interval. And this wider confidence interval is because of having a small sample size. So these dots or the studies which are close to the y-axis are said to be less precise. They do have higher confidence interval or wider confidence interval. And that is because of they do have a small sample size. That is a small sample size. And those blue dots, for example, this one and this one, you can take this one and even this one. These blue dots which are far away from the y-axis are said to be or said to have a high precision or a narrow confidence interval. And that narrow confidence interval is because they such types of studies have a larger sample size. So this is the interpretation, you know. And again, one last interpretation that we need to focus is whether there is heterogeneity between the studies included in the meta-analysis. Is there any heterogeneity or is there any outlier, extremely large outlier or extremely low outlier? So the outliers are identified by looking at whether the dots, the blue dots are outside of the confidence interval or not. So that, in this case, for example, in this Galbraith plot, we do have at least 1, 2, they're more much closer with each other, these two dots, 3, 4, 5, and 6. These 6 dots are outside of the shaded area, outside of the 95% confidence interval. That means there are 6 studies which are outliers, so that there is heterogeneity in between the studies included for this meta-analysis, So before just doing our pooled estimate, we need to treat these 6 research articles in an appropriate way. We will see that in the next videos. Another successive or series of videos will be made on this issue. So that in this case, in the Galbraith plot, here what I want to show you is that how to identify whether there are heterogeneity or there are outliers in between the studies included for the meta-analysis. So that the studies or the dots which are outside of the shaded area, outside of the confidence interval, are said to be their outliers. So because of these outliers, there will be heterogeneity in between the studies included. So that in this case we can count, there are 2 dots around here which are much close to each other, 2 dots over here, and another, the 3rd dot, the 4th dot, the 5th dot, and then the 6th dot. These 6 dots are outside of the shaded area, that is the 95% confidence interval. So that we can conclude that there are 6 research articles which are outliers. So because of this outlier, there is heterogeneity in between the studies included for the meta-analysis. So we need to just take an action on these outliers before just summarizing or calculating or the pooled estimate of the meta-analysis, So that is the purpose of this Galbraith plot, how to identify whether there is heterogeneity or outlier in the included studies or not could be detected and identified by the use of the Galbraith plot. We need to understand what does this mean first, before doing or running any statistical analysis, either in using spaces or Stata or any software. We need to understand what does this mean, what does it tell us. That is the most important thing, So hereafter we can proceed to demonstrate this Galbraith plot with the use of Stata software. So here in this slide, what I have said is already written in the PowerPoint, Okay, already, since I have already said it, I will not just read it again. In the next video, I'll proceed with the meta regression for test of heterogeneity now. So now let's proceed to the Stata software in order to just demonstrate this Galbraith plot. So I do have a data set on BCG vaccine. So there were different studies which have been done on the effectiveness of BCG vaccine on preventing tuberculosis. So that the findings of these different research works have been summarized or have been extracted by systematic review research, So now let's have a look at the data set before doing our meta-analysis. So click on data and describe that, sorry, data editor and data editor again. So here we can have a look at over here. There is a trial number of studies from 1 up to 13. So there are 13 studies included in the meta-analysis. And then the trial, the place where the studies were conducted. For example, in northern USA, in Canada, in Chicago, in UK. So the country where the study was conducted. And the authors are given here, year of publication, number of positives in the treatment group and post, number of negatives and negative in the treatment group, number of positives in the control group, positive for tuberculosis, that means, number of negatives in the control group, number of negatives in the control group. And there is a latitude measurement, that is the locational data set was measured. For example, the first data set which was done in northern USA, you see, it does have its own latitude measurement, we will use this data set in the next videos, why this latitude was measured. I will say more on that in the next coming videos. Okay, so that allocation was done by random allocation in some of the studies, alternative methods in some other methods, and systematic allocation was made in another studies, and then the study level was given in such a way, the author with the year of publication, So this is a data set that we are going to use for demonstration of Galbraith plot for this video. So now, as usual, before running our meta-analysis, we need to just declare the data set to be a metadata, So to do that, as usual, click on statistics and go to meta-analysis. Click on that. So here is an interface where we need to declare the data to be metadata, So click on setup. And since we do have a comparison group, the treatment and the control group, and then within each group, there is binary outcome, being positive and being negative. You see, negative and positive. So the outcome variable is a binary outcome between two groups. So I need to select the second circle, Compute and declare effect size for two group comparison of binary outcome. So I need to select this one. And hereafter, we need to just define the number of successes, the number of failures within the treatment group and then within the control group. So for the treatment group, the number of success is number of positives and number of failures is number of negative for the treatment group. For the control group, again, number of success is number of positive in the control group, and post C, number of failure is number of negative in the control group. Okay, effect size is in terms of log odds ratio. And click on model, and the model is random effect model. And the estimation method is restricted maximum likelihood estimation method. And click on options again, so that the study level should be selected over here. The study level is at the last one, study LBL. So click on that. Hereafter, click on submit. Now we have declared the data to be metadata, and hereafter you can see there is an additional variable which is a metadata. So now in this case we are going to just see how to do a Galbraith plot. So now in this case you can do the summary, you can do the forest plot, the ones that we have already made or done it in the previous videos, And now we are going to test whether there is heterogeneity or not. So click on heterogeneity over here. Click on heterogeneity. And be sure that the model is either a default model or random model. If you say default model, that means it is simply a random model. Or you can say random model, or random effect model, or you can say default model. Saying default model, the default model is a random effect model, So you can either tick the random effect model or the default model. So both of them are the same because the default model is random. Okay, now here as you can see, when we click on heterogeneity, what it says is there is Galbraith plot over here, heterogeneity, and then under that there is Galbraith plot, the Galbraith plot. So now we have selected the Galbraith plot and we have selected the model to be a default model, that means a random effect model. And now click on graph options in order to just edit some of the properties of the graph. Graph option, the symbol could be default, but the color, I want to change the color to be a blue color, So blue color. And even you can change the title, the x-axis, the y-axis, the legend, confidence interval plot, So you can just made as possible as many editions, So, but for this case, I want to just make the color to be a blue color. So now click on OK, and then click on Submit. Yeah, this is the one that we are observing in the PowerPoint, As I have said before, in the PowerPoint, this horizontal line is no treatment effect line, which is indicated over here in the footnote. I see, and then this slope is a regression line, the overall effect of a BCG vaccine on tuberculosis. So that is the regression line. And then the dots are study-specific effect sizes of BCG on tuberculosis, The effectiveness of BCG vaccine on preventing tuberculosis in each of the studies, has been already reported over here. And then those dots or blue dots which are near to the y-axis are having a less precision. That means wider confidence interval. and that is because of they do have a smaller sample size. And those blue dots which are far away from the y-axis are said to be more precise. That means they do have narrow confidence interval. That means they do have what? A higher sample size. The precision is related with the sample size. The more the precision, the higher the sample size, that means. Or the smaller the sample size, the wider the confidence interval will be. And this, the shaded area is a 95% confidence interval for this regression line. And we expect if there is no any heterogeneity or if there is no any outlier between the studies included in the meta-analysis, we expect all of the dots to be within the shaded area, But if there are dots or blue circles which are outside of the shaded area, that indicates those dots are outliers. And because of these outliers, there is heterogeneity between studies included in the meta-analysis, So that in this case we can conclude that there is heterogeneity. There is heterogeneity. Okay, now again I can add another addition over here in order to just add labels to markers and the variable to be the trail, the number of trail. The trail, that means the trail has been listed as 1, 2, 3, 4 and up to 13. There are 13 trails. Because I am just interested to know which trail is an outlier. In addition to see or looking over there whether there is an outlier or not, which specific study is being an outlier. I want to know the specific study which is an outlier. So in order to see that, I need to just click on graph options over here, and then over here you need to say add labels to the markers, and in front of the variable click over there and then the trail. So the trail number will be displayed on the graph, Click on OK and then submit. You see, now in this case, in addition to just the blue circles, there are numbers attached with the blue circles, So this is the number 8 trail, which is an outlier. This is the 6th trail, which is an outlier. This is the 4th trail, which is an outlier. This is the 10th trail, an outlier. And here there is the 2nd trail and the 7th trail. So that from here the 7th trail, the 2nd, the 10th, the 4th, the 6th, and then the 8th trails are outliers. So in order to just identify which specific studies are outliers, we can just modify the graph or edit, in such a way. So with this approach, you can assess whether heterogeneity because of outliers, and you can see which specific studies are outliers by using this Galbraith plot, So this is all what I have to show you for this Galbraith plot. And lastly, please let me remind you to subscribe and share and like my YouTube channel for others, which could be benefited from this content. So now I will proceed to other tests of heterogeneity with a coming series of videos. So for this time, this is what I have for you. Thank you very much.